our webinar organized jointly with Decode Science, our distributor in Australia and New Zealand. Regarding the organization of the webinar, the first part will be held by Joshua from Decode Science, who is going to introduce Decode Science as our distributor for Australia and New Zealand. And in the second part, I will present our company, and I will also perform a demonstration of our tools and services for interpretation of NGS data. Then in the last part of the webinar, we will discuss your questions. So please feel free to write your questions in the Q&A section. And also for your information, this webinar is going to be recorded for your future reference. So now, Joshua, over to you, please.
Yes, this is Tomas, but I see Josh 3 times. I'm not sure which one.
I think he's now connecting. Hi, Josh.
Beautiful.
Now you are online, Josh.
Great. Sorry about that, everybody. So Decode Science is an Australian-based distributor focused on genomics, and our mission is to help clinicians and researchers around Australia enable discovery by supporting some of the best international brands around the world. So we currently represent MGI for sequencing, Swift Biosciences, Swift Bionanogenomics, and we've just started working with VarSome in the market. And I will pass it back to the VarSome team to explain how they can assist you in the clinical space.
Thank you, Josh. I'll share my screen again. All right, so here. So Safiter, our company, is a precision medicine and bioinformatics company. It's based in Switzerland, and we are on the market since 2014. And we develop tools for processing and interpretation of NGS data, specifically for clinical purposes, although we also serve research clients. I would like to start my presentation by bringing your attention to the challenges we face when bringing NGS technology to the clinical practice. There are many challenges out there. However, from our perspective as a precision medicine and bioinformatics company, we focus on 3 challenges in particular. The first challenge is the fact that the annotation data is fragmented and spreads over many data resources. I like to say that the annotation process of data is like a puzzle game. So to get a complete picture, you need to place all the pieces of the evidence in the right position. Then the second challenge is that our understanding of the mechanism of diseases is becoming better every day. And therefore, there is a constant need for re-evaluation and for keeping the annotation data up to date. And then the third challenge is the lack or the little standardization and consistency when it comes to the interpretation of pathogenicity. For example, there might be 2 laboratories interpreting the same variant differently simply because they have access to different annotation data resources. These 3 challenges are the reason why we have created VarSome, which is the free and open knowledge base and community. I believe some of you might already know the platform. So VarSome aggregates and cross-references annotation data resources. We pull the data from over 70 data resources and bring the data in a single place called VarSome. In other words, these are the 70 pieces of the puzzle game. So we have annotation data resources such as NOM-ID for population frequencies, ClinVar, expression profiles, annotation data resources for cancer samples, like ICGC, PMKB, CIVIC, COSMIC, in silico predictions, conservation scores, clinically relevant data for genes, phenotypes and diseases, clinical trials, drugs, list of publications, and so on and so on. Over 70 data resources in a single place. And for challenge number 2, we keep the data always up to date. So you can always find the latest annotation data from all these over 70 data resources. And we implement guidelines for standardization of the interpretation process. You can find interpretation of pathogenicity based on ACMG guidelines. And we are also implementing guidelines for somatic interpretation, such as AMP, NCCC, or ASCO. And actually, AMP is coming in the upcoming weeks. However, VarSome is not only about the annotation data resources. VarSome is also about the community around, about you. There is a large global community that contributes back to the VarSome knowledge base. So you can find publication links from our community, or classifications, comments from VarSome users. And basically this creates an additional layer of annotation data, which is unique to VarSome, and you can leverage it for your daily work. So this is the very quick story of the free platform and community around it. And now I'll present VarSome Clinical. So on top of the free platform, we have developed VarSome Clinical, which is a professional platform for the interpretation of NGS data, starting from FASTQ or BCF. VarSome Clinical is certified as an IVT medical device, an in vitro medical device, which means it can be used indeed in clinical settings. Our platform is HIPAA compliant, which is relevant for our US-based clients. Our company is also GDPR compliant, and we are certified with ISO certifications for data quality, and we are now also becoming certified with ISO for information and data security. In addition, we are based in Switzerland, which is a country that is very sensitive on data security and data privacy. And we need to comply with a number of local laws in terms of data privacy and data security. So basically your data is safe with us and we don't do anything with it. So VarSome Clinical shares the annotation knowledge base with VarSome, the free platform. With the free platform, you can look up individual variants. However, with VarSome Clinical, you can start directly from FASTQ or BCF. And at the end, you get a clinical report, which is completely customizable. With VarSome Clinical, you can process any type of NGS data as long as it is human DNA. You can process gene panels, off-the-shelf gene panels, or your custom gene panels, or exomes or genomes. We support both sequencing platforms, Illumina or MGI, and you can also your own BCF file as a result of the secondary analysis. So here I would like to highlight where the challenge is. The challenge lies on the interpretation part. You need to get as many pieces of the puzzle together as many pieces as possible to get a complete picture. So VarSome Clinical covers all the steps of the pipeline, the primary, the secondary, and the tertiary analysis. However, our main focus lies on the interpretation part. This is because we believe root alignment and variant calling is relatively simple compared to the interpretation part. So while the first step of the data treatment is primarily automated, the variant interpretation must be performed manually by genetic analysts in many cases. Therefore, the variant interpretation implies comparing multiple databases, evaluating potential artifacts, prioritizing variants, and selecting the one that is responsible for the patient's condition. And therefore, VarSome Clinical has been designed with this aim of facilitating the interpretation part of the analysis. So you can process your NGS data with VarSome Clinical in 4 steps. First, you have to upload your sequencing data to our servers. And once you have uploaded the data, you can run the pipeline. So VarSome Clinical offers a wide range of pipelines covering pretty much all the clinical use cases. We have pipelines for germline samples, somatic samples, also pipelines for de novo variants in triome, structural variation. We also include a pipeline for UMIs, unique molecular identifiers, allowing detection of low frequency variants typically found in somatic samples. VarSome Clinical comes with a website interface. It runs on our infrastructure, and you can access it over the internet as if it was a regular website, which means that the platform is always updated. You have always the latest annotation data available, and you do not have to worry about the security or the scalability issues. Basically, we take care of it, and you don't need to install anything locally. In the end, you may generate a clinical report over which you have complete control. You need to set up your template specifically for your workflow, and you can use it for all your analysis. The template can be fully branded according to your institutional branding policies. So VarSome Clinical is not a replacement for your in-house bioinformatics expertise. On the contrary, it complements your bioinformatics expertise. VarSome Clinical is a productivity tool which allows you and your bioinformatics team to work more productively and systematically, and to focus your energy where it is really needed. And basically together we can increase significantly the diagnostic yield for your patients. So here I just wanted to share with you very quickly our list of clients. So you can see, as a company based in Switzerland, we have clients pretty much in every European country. And we have also a number of clients in the global scale in the US, Asia, Latin America, and so on. So VarSome Clinical, when it comes to pricing, VarSome Clinical does not have a license. There is no commitment. VarSome Clinical charges on a per sample basis. When it comes to the FASTQ files, we charge on the number of megabases in bits per sample. But also for clients through distributors, we may also charge on the basis of target regions, which typically results in better price. And for BCF, the price depends on the number of variants. So feel free to contact Decode Science, our distributor in Australia and New Zealand, to get the exact pricing sheet. Okay, so there are basically 2 ways how you can use VarSome Clinical. You can use it as the so-called stand-alone solution, which is open for any type of NGS data. It could be gene panels, exomes, genomes, commercial gene panels, custom gene panels. You can upload anything you like, and we charge on a monthly or quarterly basis depending on the usage of the platform. It is a simple pay-as-you-go model. There's no commitment and no license. The other option is the so-called bundled solution. It is a form of prepayment, and it includes the kit for the library preparation bundled with VarSome Clinical for data interpretation and reporting. Basically, it's a form of prepayment which allows you to analyze a certain number of gene panels, for example. We also offer on-prem installation, which normally makes sense only for large diagnostic centers and large hospitals that have a large volume of samples. We work with distributors. In Australia and New Zealand, we collaborate with Decode Science, who is very well familiarized with the platform, and they may be able to answer your basic support questions or questions about the use of the platform. So this is my quick introduction. Now I would like to give you a very quick introduction to VarSome, the VarSome Clinical interface. So for that, I'm going to stop sharing the presentation. Okay, can you see VarSome now?
Yes, Anna, it's all right.
Thank you. Okay, so here you can see the free platform available at varsome.com. I believe some of you, at least some of you, have seen it before. So it is the knowledge base for annotation purposes and the community around it. Here, basically on a single page, you can see a wealth of annotation data coming from all these 70 data resources. So I'd like to tell you a little bit more about the interpretation procedure based on the ACMG guidelines. So in this particular example, this is a variant in a BRAF gene in which you can see 3 ACMG criteria that fired automatically based on the aggregated evidence coming from all these 70 data resources. So here you can see the explanation for every criterion, telling you in a great detail why it fired. Basically, this allows you to review every criterion and possibly make your own judgment call. Not all the criterion can be fully automated. Basically, the ones in which this little square is empty. That's because certain criteria require patient-specific information or information, for example, information about the allele segregation. In those cases, it is you who is supposed to trigger or untrigger certain criteria in line with your best knowledge. You can also specify the transcript for ACMG-based classification, which may affect the final verdict. And basically, you can reclassify also the strength. And you may also specify the strength of the evidence in line with the ACMG guidelines. So clinical laboratories rely on Climber a lot, which is a very useful resource. However, according to the ACMG guidelines, Climber is only a supporting piece of evidence. So again, this is about the puzzle game. we should not rely only on 2 or 3 pieces of the puzzle game. With 3 pieces, we cannot get a complete picture, and we need to adopt a more sophisticated approach, especially when it comes to larger data sets as exomes or genomes. So now when it comes to VarSome Clinical...
Anna, excuse me, we have a question here from Chris. How does VarSome Clinical ACMG classification compare to the free VarSome ACMG classification? So, Chris, there are certain differences, especially in terms of additional criteria which are evaluated in VarSome Clinical, and these additional criteria are always related, to a patient specific information. So there is a slight difference.
Thank you, Tomas. Can you see VarSome Clinical now? Just to make sure I'm sharing the right page.
Sure.
Okay. Okay, so VarSome Clinical shares the annotation knowledge base with VarSome, the free platform. However, it allows you to start directly from FASTQ or BCF. First, you upload your data. You can upload your data directly from your computer, but we also have the API available, so the application program interface. It's available for every step of the analysis. So basically, you can automate the whole workflow. And here, once you've uploaded your data, you can start the pipelines. The launch analysis. So we have 2 more normal pipeline for trios, carrier risk screening, structural variation, pipeline for UMIs, pipeline for copy number variations, and so on. And assuming that we start from FASTQ, we should specify the details of the pipeline, whether it's a germline or a somatic sample. We should specify the assay used for the library preparation, which could be basically any gene panel. Commercial panels are already available on the platform. You should also specify the reference genome. And you can also type the patient phenotype and diseases in here on the left, so that later in the process you can use these terms for the variant filtering. And then you can start the pipelines and the analysis. So the analysis of a whole exome takes about half an hour. For gene panels, it would take between 5 to 20 minutes or half an hour, depending on the size of your data. So here I would like to show you very quickly the results for a whole genome analysis starting from FASTQ. We have almost 5 million variants. And basically, as you can see in the variant table, the variants are classified by default. They are ordered according to the ACMG, according to the pathology, according to the ACMG guidelines. And then around the variant table, you can see a wealth of annotation data broken down in tabs. Basically, you can see the region browser, ClinVar, somatic data resources, clinically relevant data resources, expression profiles, in silico, prediction scores, conservation scores, and so on and so on. Also, VarSome Clinical allows you to, this is a very powerful tool, so it allows you to set up your own costume classifications. So for example, this variant, the first one, it's pathogenic according to the ACMG guidelines. However, if you don't agree with the default verdict, you may reclassify the variant as benign in here, as benign or uncertain significance, for example. And you can even set up your own costume classification. For example, you could say sample that needs, variant that needs to be discussed later. So here in this other example, you can set up your own costume classification. This one would be the one I created, needs to be assessed later. And you can also associate that with a certain variant. One of the powerful things about this tool, the custom classification, is that these custom classifications stay with the variant, which means that once you upload a new sample with the same variant, you will see the same classification mark. Also, we can take your manually created list of variants. Typically, you may have it on a spreadsheet. And we can add it to your account with VarSome Clinical for the purpose of variant classification. Another really useful feature is the sample cross-referencing. So this is basically something that allows you to go back and forth between your past cases and your current cases and compare the allelic balances, the patient phenotypes, together with your costume classifications. And over time, it helps improve your productivity and, more importantly, the diagnostic yield for your patients. So in this example, we see this particular variant that is selected has appeared in VarSome Clinical database in heterozygous form in 55 other samples. And here, this is my demo account, but once you start uploading your samples here in the bottom, you would see if you have other samples for your group with the same variant, and you would have a link to go to those samples, and it will help you compare samples from the past with your current samples. Then here in the analysis actions, you can see that the platform comes with a number of QC reports. You have the coding coverage report, the region list coverage report. Basically, you can download this coverage report. You also have the BAM file, the BCF file. This coverage report, you can download it in an Excel sheet that it's broken down by individual genes, exon number, and sequencing depth. Basically, this would be the place where you can identify those regions that have low coverage or regions that have been skipped altogether for some reason. And this sheet allows you to troubleshoot the issues with the assay design or issues with sequencing, so that it can be useful, especially when it comes to custom gene panels. And it allows you to optimize the whole workflow. Then when it comes to filters, in here, let's create a new filter. So you can see that there are quite a few filters. You can filter your variant based on a number of criteria, the ACMG criteria, frequencies, chromosomal position, pathogenicity, zygosity, function. And you can also set up a gene list, and you can use this gene list as a filter. From a pricing perspective, we charge you only once. We charge you for the initial analysis, and then you can apply all these filters, including a gene list. So basically you can have multiple gene lists, for example, for an exon, and they are all included in the original sample price. So here, let's create an example. Let's say we want to filter sorry, those here, we want to filter those variants that have a frequency that is less than 1% and that have a pathogenicity, those that are pathogenic and likely pathogenic. Then we need to put a name to the filter. And we save the filter. So now here we can see the number in green is the number of variants that have been filtered out. And the number in blue is the number of variants that remain after applying a certain filter. These filters can be activated and deactivated. That's why they are called the dynamic filters. And we can also edit the filters by clicking here. We could add, for example, here, and we could add other criteria in the filter set. So it's important to know that the filtering works very fast. We have built a whole platform keeping the performance in mind. So we are left here only with 5 variants down from 5 million variants. Basically, these filters are dynamic, and you can play with these filters, and the results update dynamically. Then we also have even more comprehensive filters, which are called the algorithmic filter. These algorithmic filters are basically little programs that we build specifically for our customers. So we like to be in a close touch with our customers in order to be able to provide as much personalized service as possible. And these filters are part of this story. So for example, with these filters, you can analyze de novo variants in trios, carrier risk for couples, compounds, heterozygous variants, segregation kind of analysis, and so on. Basically, we can encode any kind of filtering that you can possibly think of. You just need to tell us how you want the filter, how you want to filter the variants, and we will develop the filter. And the use of these filters is free of charge. There might be an initial development. The initial development might be subject to charge. So then when it comes to reporting, so once you've narrowed down the list of variants, you need to decide whether you want to report it or not. Once you make the decision, you select it for export, and then you can proceed and generate a report. You can download the variants on a spreadsheet, or you can generate a clinical report right on the platform. And basically, you have complete control over the report. First, you need to set up your template, and it is completely customizable. So you can customize it according to your branding policy. You can, for example, here you can upload your own logo, you can set up a header, you can change the color, the font, the size. You can even include pictures and tables. And then here on the left side, you get the list of the widgets. In other words, these are the pieces of the annotation data that is available for a particular variant. And the way it works is basically that you can drag in those widgets that you want to report, that you want to include in this report. So you may drag in variant information, information on the gene, the list of drugs. You have also information about the clinical trials, etc. And everything is fully editable. Let's see. Well, everything is completely editable. So if there's something that you want to remove, you can remove it. And if there are comments that you want to add, then you can add it as well. And then in the end, you can save it as either as a, here in the top, you can save it either as a PDF or as a document. So this has been a very quick introduction to VarSome Clinical. Basically, it's a complex platform with a number of sophisticated features for custom classification, sample cross-referencing, and so on, which allows you to increase your productivity and diagnostic yields at the end of the day. Basically, what we want in the end is to improve the outcome for the patients. Just to wrap up, VarSome Clinical is a complete solution for any kind of NGS data, gene panels, exomes, genomes. There's a wide range of pipelines. And there is no commitment, there's no license. We charge on a per sample basis. The platform is certified as IVD in vitro diagnostic medical device. So thank you very much, and I would be now very glad to take some questions. Also, Tomas, our head of business development, is here to help with the questions that you may have.
All right, thank you, Anna. Indeed, we've got a number of questions from the audience. So Anna, I'll be asking you these questions. So the first question from Bing. Can we process data from Swift Biosciences?
Yes, so Swift Biosciences is actually one of our partners. So yes, you can upload the data from Swift Bioscience kits.
Yes, indeed. You can upload any kind of NGS data. Either your custom gene panels, commercial gene panels such as Swift Bio. Exomes, genomes, the platform is open for any kind of a human NGS data. Having said that, we currently don't have a pipeline for CT DNA. However, I believe it's also under development. However, we cannot comment on delivery date yet as of now. Do we have a server in Australia?
So we have a server in Switzerland, data center there. And there's also the option, we also have the cloud in the US. At the moment, we don't have a server in Australia. I don't know, Thomas, if you want to add anything on that.
I think we can add that's something that we're working towards. We understand that it's a requirement for clinical samples. So our goal is to have that service in place in the near future.
Yeah, exactly. So we are basically now discussing the installation of our tools locally in Australia in collaboration with Decode Science. Do we have a pipeline for CNV?
Yes, we do indeed. So let me show you what it would be. So here in the launch analysis, you also have the section for your copy number variation and structural variation analysis.
Yes, yeah, of course, the CNV pipeline is also available, including a pipeline for structure variation. Another question from Harry. Is it possible to import custom parameters in a VCF file and then create filters using those custom parameters? So yes and no, it really depends on the way we look at it. basically we can process any VCF file with all the custom parameters in there. However, the filtering based on these parameters, it's not available out of the box, but it needs to be delivered as a customized algorithmic filters. Yes, so yes, it's possible, but it may require certain development on our side in the form of a specific customized algorithmic filter for you. What about AMP criteria and guidelines, Anna?
So yes, AMP criteria we are currently developing and we're gonna implement them. So it's not currently implemented yet, but in the very near future, I think in the upcoming weeks, we're going to implement them.
Yes, so AMP guidelines are basically just around the corner. How do we deal with somatic mutations? I think your question is probably referring to the interpretation part. If that's the case, we also provide access to multiple somatic annotation data resources such as Cosmic, Civic, PMKB, TP53. Now, another question from Anthony. How do we deal with versioning of our pipeline? If you change your secondary analysis pipeline, a diagnostic laboratory will need to re-verify the workflow. Yes, so the analysis it's basically frozen in the time, in the moment of running the analysis. On VarSome, on the free platform, you have always the latest annotation data. However, in VarSome Clinical, the annotation data are frozen in the moment of the analysis. And eventually you may re-annotate your sample at some point in the future, if you decide so. And later, using algorithmic filters, we can highlight only the variants where the classification has been changed. Regarding variant callers, we don't develop variant callers in-house. We implement variant callers and aligners from Sentient. I think we have covered all the questions from the audience, Anna, for now.
There's just one more that came in. Will the AMP classification be available with VarSome Premium?
Yes, so VarSome Premium, thank you for bringing this up to our attention. So indeed, VarSome Premium it's the latest addition to the product portfolio. VarSome Premium, as it sounds, it's a premium version of the free platform, which gives you access to licensed and proprietary annotation data resources. So on the free platform, you can find annotation data which are publicly available from the public domain. And VarSome Premium, you get access to additional third-party licensed data resources. And you also get access, to premium features such as AMP-based classification, which is available will be available very soon, both in VarSome Clinical and VarSome Premium platforms.
And then lastly, we have a last question that says, what do you think is your advantage over other competitors, for example, Sophia Genetics?
A good question. Our focus has always been the interpretation part of the pipeline, especially when it comes to larger data sets such as exomes, genomes, or large gene panels. What I would also say that our platform is also customizable through algorithmic filters. We like to stay in close touch with our clients and provide as personalized services as possible. And that's for now what I can say about our competitors. But perhaps later we can schedule a direct call, Anthony, with you, and discuss details offline.
Okay, so I believe that was the last question for today. Thank you everyone for attending the webinar and for all your questions. As I mentioned before, it was recorded, so we're gonna send it by email for your future reference. And as Tomas just mentioned, feel free to schedule a call with us, or feel free to contact Josh from Decode Science in case you would like to have further information and further conversations, and in case you have any other question.
Josh, do you have some closing remarks?
Nothing specific. Thank you everyone for attending. And if you do want to have further in-depth discussion, reach out to us and we can schedule more details, or alternatively, we can provide you access to a demonstration account where you can explore the power of VarSome Clinical yourself. So just let us know. Thank you once again.